Thank you, everyone. Thank you for coming to my talk. My name is Juliette. I'm a PhD student, and today I would like to present you my work about genome streamlining and how mutation rate and population size can drive genome streamlining, also known as just genome size reduction. So first, a bit of context. We know a lot of different bacteria species which have a lot of different genome sizes and coding densities. And even if we focus on streamlined bacteria, which are bacteria who underwent a recent genome size reduction, and that are all the color points here, we see that there is a huge diversity in sizes and coding densities. There are a few hypotheses in the literature to explain that, and they focus on population size and mutation rate, but it has not been thoroughly tested because it's very hard to do in vitro and in vivo. So we want to try to test these hypotheses with simulations. You do not need to know everything about our simulation methods, but you do need to know a few things to understand our results. First, we have an explicit genome, and on that genome, we are able to recognize RNAs and genes, and so we can decode any sequence into a phenotype, hence a fitness, and we can know perfectly which part of the genome are coding or non-coding. It's an individual-based model, and we have selection on the fitness, and the mutations are occurring on the sequence. And so we modify the sequence, and afterwards we compute the fitness. There is no a priori fitness distribution effect. Then we have an experimental evolution approach, which means that we use a pre-evolved organism, and then we confront them to different parameters. So we will confront them to changes in population size or genome rate, population size or mutation rate. So first, if we look at the effect of population size, here we have our control experiment with a standard population size, and we multiplied it either by 4 or 16, and we have the change in the coding and non-coding DNA. So what you can see is that population size doesn't have an impact on coding DNA, but it does reduce non-coding DNA. So that means that population size can drive genome streamlining. Similarly, for our three different mutation rates, mutation rate can also drive genome streamlining, but in a very different ways, because here, both the coding and the non-coding part of the genomes are impacted. And so if we combine these different types of effects, and if we look at a lot of different population sizes and mutation rates, we see that we can reach very different genome sizes and coding density. So for example, starting from our original population size and mutation rates, we can reach the same coding density by changing the mutation rate or the population size, but with very different total sizes. So that's observation, and it can explain the diversity of sizes and coding densities we have in vivo, but that's just observation, and the next step was to try to understand why it is the case. So we do have some mechanistic explanation for that, but first I need to tell you a little bit about robustness selection. So what we call robustness in our model is the replicative robustness, and it's the probability that your offspring is as good as you are. So here, for example, we start from a standard genome with a good fitness, and if it undergoes a duplication in the non-coding part, then the fitness remains the same, but then the grandchildren of our original sequence have more chances to have mutations because the mutation rates are per base, and so the robustness has decreased. Whereas on the other hand, if we remove some non-coding bases, we keep the same fitness, but we increase our robustness because then the grandchildren have more chances to be as good as the original sequence. So we did measure both fitness and robustness in our simulations, and here it's the time series of our data for robustness and fitness in our experiment where we multiplied the population size by 16, and so we can see that both increase, which is quite expected because increasing the population size increases the efficacy of both types of selection. So we do reach better fitnesses and robustness. And it's very different what happens when we look at the increase in mutation rate. Because when we increase the mutation rate, we suddenly have a huge drop in robustness because all of a sudden you have a lot more mutations than expected. And that is followed by a drop in fitness because you have a Müller-Rachet effect. Since you cannot transmit your information faithfully, you cannot select it. And then we have to regain first some robustness, at least, to be able to regain some fitness and to have effective selection again. So in both cases, we have robustness selection, but it's very different because in the first one, it is secondary to fitness selection. We keep our fitness and we just increase robustness in addition to that. Whereas in the second case, we have robustness selection, which has to come before fitness, because you cannot select information that you cannot transmit. And that explains the results we have, because here is our genome structures in both cases. And so in blue, you have the total genome size, in orange, the coding genome size, and the difference in between is the non-coding genome size. And when we have just a huge population size, we reduce the non-coding genome size because that's a way to select for better robustness. But we cannot reduce the coding genome size because we do select for a good fitness. So you cannot remove half your genes, otherwise you won't be as good as before. And if we look at what happens in the mutation rate, we see that we lose first both types of genomes. So we lose coding bases and non-coding bases, because the only genomes sequences that will have grandchildren in the end are the ones who lost sufficient amount of genome to be able to transmit their information again. But they do not lose both coding and non-coding bases in the same proportions, because among the sequences which have lost the most sequences, the one with a better fitness are the one who kept more genes. So both types of genome structure can be explained by robustness selection. It just depends on the relation between robustness selection and fitness selection. So if you want to know more about that, there is a preprint which is already available on bioRxiv, and we are also developing a mathematical model to explain these results more thoroughly. And thank you for your attention.
I'm coming like an audience, not online questions. Very interesting talk. Thank you. My question would be, do you know if the gaining of fitness can be also connected to additional genome rearrangements which occur during losing and coding parts of the genome?
We can reach very high fitnesses with either very short genomes or very long genomes, but when we start from pre-evolved genomes, it's mostly correlated to having longer or a little bit more genes, because you can encode more informations. So you can have rearrangements, but on average, you do have to maintain the size of your coding genome to get a good fitness.
Okay, thank you.
Thanks.
So could you extend also your model to also include some changes in the fitness landscape, that suddenly some changes, the previously fit organisms would not be fit anymore and others would become suddenly fit to some dramatic change in the environment?
Yes, we can do that because our sequence is decoded into a phenotype, which we then compare to an environment to get the fitness, but we are able to change that environment. So we can make it more complex or just a little bit shifted and explore different impacts of environmental changes. So that has been done, not by me, but by other people in the team previously. And there was also a response in genome size to different environmental changes.
Very interesting, thank you. Let's thank our speaker again.